Hello, everybody. Good evening. Welcome. Colleagues, guests, friends, and most importantly, family. Thank you, everyone, for being here to celebrate the inaugural lecture of Professor John Tregoning. I'm going to start off with an information announcement. There is no planned fire drill. So if anything were to happen of that nature, please follow the signs and move outside to the assembly point, which is on the corner of Exhibition Road. Okay, I'm announcing that this event is being recorded, so welcome to our online audience. We are going to have a wonderful lecture from John, I know, today, and at the end of that, John has agreed to take some questions, so I will be chairing that session, and the online audience are also very welcome to post questions, and we will take those as well, at the end of which I will hand over for a sum up, a vote of thanks, and then we'll all have a lovely glass of wine. Just before I hand over to John, I just want to say a very brief few words about him. It turns out when one looks at his CV that he has been hanging around Imperial College for a very long time. Since 1999, apparently. I'm not really sure quite what to think about this. Either we just couldn't work out how to get rid of him, or he loves us very much, or we love him, but I think it must be a good thing. He is indeed a wonderful colleague. You can see there are many people in the room who have known him actually even longer than that, apparently, and are still here. So that must say also something about John and his friendships. Very briefly, I would just like to say a huge thank you to John. Since I became a head of department, the one thing that really strikes me about John, he's going to give us a wonderful tour of his science today. I'm not going to say anything about his science. You're going to hear all about that. But John is just one of those people who does the right thing. And I am so very, very grateful to John, who has been a moral compass for me throughout the last 6 years of trying to act as a head of department. If I'm doing something wrong, he will call it out and he will do it right. He will help me word things. And not only that, but he will check in on me at the end of a week and say, how are you doing? And there's not many colleagues that bother to do that, but John always does. And I just think that sums him up absolutely. He is somebody who does the right thing, he knows what's right to do, and he really cares for people. And you can see that because he has lunch and coffee with his trainees, and everybody's smiling and happy, and you can tell they adore him. And so I am very, very grateful to have John as a colleague. I'm very, very proud that he's here to give his inaugural lecture. And I would just like to say, scientist, author, media star, and now Professor Tregoning, over to you.
Thank you. Thank you for those very kind words. It feels slightly strange not introducing myself, but if you've not yet figured out who I am from the cardboard cutouts, massive photo of me, and Wendy's introduction, you might be in the wrong place. It's a huge pleasure and privilege to be here tonight, and thank you for the kind words, and thank you all for coming, especially those also to those joining us online. And thank you for being part of the journey with me. The next bit might be a bit Oscar acceptancy, but here's some ground rules about what I'm going to say. The first is, it's okay to laugh. I've sometimes found I need to say this during science talk, or at least that's my excuse when the jokes don't land. If you do get bored, count how many pictures I have put of myself in it. There should be some real events, and there might be some science. I've been lucky enough to work in some great labs in my time, and it's thanks to Peter, Gordon, and Robin, which Nixon, Openshaw, Dugan, and Shattuck, respectively, I'm here talking today, more of which to come, talking about my science. But not to downplay the role of all my fabulous colleagues that I've worked, past and present. One of the big joys of the last year is that we've moved departments and we've physically moved to a space where we bump into each other all the time, and the kind of tea room chats that we have is really, really nice. And it's lovely to see so many of you here today. I owe a huge debt to all the wonderful people who've wielded pipettes to answer all the random questions I come up with. There's certainly a bit of a process to how the lab works, maybe not necessarily unique to my lab. I tend to throw 100 random ideas at my students, and they say, yes, boss, we'll definitely do that, and then carry on doing the thing they were already doing, ignoring me entirely and hoping that I'll forget. One of my recent postdocs describes this as science vomit. The ideas just bubble forth uncontrollably. We're now entering the 4th iteration of my lab group, and it's a source of great pride that my 10th and 11th PhD students, Hadi and Meryl, have just submitted their theses. And I especially want to thank Livia and Valeria tonight because they're fielding the online questions, so I thought I'd better say nice things about them too. Science is a team sport. According to one internet data, I've written papers with 559 people. I'm not going to be able to read through all their names today. And I want to thank all of them for sharing ideas and resources and time. And as you can see, there's a top 10 of repeat offenders, or frequent flyers they might be described of, who I've published with many, many times, and I'm particularly grateful to them. But my team and I wouldn't be able to do the science we do without everyone else around us. There's people who keep us safe, who get us paid, who fix all the stuff we break, who look after our mice, who help us to teach, who share our ideas to wider audience, and generally just make things happen. The academics often seem to get the limelight, but without everyone, the whole system doesn't work. So I'm completely grateful to everything they do. And if I've not included your picture on there, it's not because I didn't remember you. And finally, none of this work would be possible without grant support. So this comes from a mixture of charities and the government. So if you're a taxpayer, I salute you. Don't worry, it's not all going to waste. Apparently, for every £1 you invest in my lab, £1.20 return comes to the British government and the economy. So that's great news. But that's the thanks done. Let's move back to the thing you're all here for, to hear about me. It's common at this point to paint a glowing, glitch-free career portrait. Mine goes like this. Undergrad at Cambridge, PhD at Imperial, postdoc, lecturer, author, professor. As you can see, at no point have I ever not looked cool. In fact, I think the lecturer photo is a real highlight, my science tramp phase. But my actual road has been a little bit more lumpy. The BA was a lot of fun, possibly too much fun. Actually, I found myself surrounded by people much cleverer than I and much more willing to work hard. And it took a while to adjust to that process and really possibly didn't do as well as I should have done. I graduated. I genuinely had no idea what I was going to do next. I sort of ended up doing a PhD by accident. Nothing works for the first 12 months. I really very closely chucked it all in. I was completely frustrated. Don't take that as a lesson. Finally, I flipped over to moving to America. I'll talk about it later, but things started to work very well. I then finished my PhD. Again, had no idea what I was going to do next. But then I finally got a lecturer post, but then everything got rejected, and then I had a book. So there's always ups and downs. I think the narrative of my life is much more complicated than the version that I might sell on my CV to get promoted. It's traditional at this point in the inaugural to show how well-traveled you are. And at the very zoomed-out view, it looks that way. So America, the UK. However, my career actually looks like this. PhD in South Kensington, postdoc in Paddington, first lecturer job in Tooting, then back to Paddington. At this rate, I'll be back in Paddington in about 5 years' time. Those of you who are really eagle-eyed will notice that the PhD and the professor jobs are slightly separated. That's because I've moved one building to the left. And my lectureship and my postdoc in Paddington, I'd moved up one whole floor, so I'm quite adventurous. I'd also hoped there'd be some kind of blue plaque or recognition of my time in biochemistry. In fact, my old office is now the cleaner's cupboard. Noticeably, they haven't even punctuated it right as a kind of double blow. So how did I get here? Ooh, don't turn the page. Luckily, there's a 30-second delay in case I say something naughty. Let's begin at the beginning. This is my family. We're looking peak 90s here at a family wedding. My parents are not particularly scientific. I hope they won't mind me saying that. My father, Richard, has invented an entire new branch and wholly wrong branch of science called dad science, which focused on something called the pickled onion gene. My mother, Georgina, is more mathematical, and her father, in turn, was a civil engineer, so there's a bit more STEM in that side. My sister split science and history. My older sister, Emily, was also trained as a civil engineer, and my younger sister, Claire, developed an offshoot of dad science called Claire Science. Different, but equally wrong. I mostly grew up in Berkhamsted, north of London, and I'm not actually the first member of my family to work at Imperial. My mother worked for the rector, Lord Penny, in the Byte building back in 1967. You may be surprised to hear that I was socially awkward at school. But I discovered several of my lifetime loves there: the army, running, reading, drinking, and science. One of my lasting influences was the encouragement to experiment. At primary school, we had a teacher called Mr. Pickle, who allowed us to throw sodium into puddles, What the learning objectives of this was unclear, but it was terribly fun. And then at Haileybury, amongst other things, there was the Wine Society. This was hosted by Dennis Cruz, who's kindly in the audience here today. Each week we tested wines of various qualities and made notes, a form of experimentation, I suppose. One of my friends reminded me, she said, I hope you've grown up a bit since one of your key questions during chemistry of whether farts smell more in the bath and why. All of which led me to Downing College, Cambridge. I'll admit this was probably not my most academic period of my career, due to a combination of things. Opportunities to do things that weren't just academic, moving from a school system where you're told what to do and when to becoming an adult learner, with a dash of no longer being top of the class without trying. There were some highlights though, writing an essay about pigs with being able to fly from the perspective of the pig, being quite bad at a range of sports, and switching a journal club paper over so the whole class learns about watermelon flesh colour genes instead of fruit flies. I began to refine my way scientifically in the third year when I did my part 2 in genetics. This was in part due to Dr. Phil Oliver, who's pictured here, who wrote my first ever academic reference. He said, it was one line, he writes quite well and is quite funny, which I think still holds true. But the key person at Cambridge for whom my science career was really kind of launched was Dr., who's now Professor Stephen Russell, the head of Department of Genetics. I was placed in his lab for my final year project, and due to the ludicrously short terms at Cambridge, this ran to a whole 8 weeks. But a vital thing happened during that 8-week period. We were looking at some fruit fly salivary glands, and that's what this kind of black blob is reproduced on the screen. And Steve said to me, you're the first person in the world ever to see that thing. And it was that was enough, that moment of like discovery. It was so exciting. And that's something that hopefully the students and people who work with me get occasionally, that in amongst all of the things not working, occasionally something does work. And that thing is so exciting. And whilst that seed was sown, it took a while to germinate. On graduating, I moved to a commune in the woods in New Jersey and taught climbing, kayaking, and other outdoor skills. The whole ethos of this place called Project Use was about education and learning, and this really led me back to the UK to apply for a PhD. And I think this was a key tipping point, moving from you go from being a consumer of sciences to a creator of science, and that kind of process has been really exciting. Having decided to do a PhD, the question was where? Back when I was applying, Alistair says, you're so old, you have to say something about it all being fields around here. All of the PhD in the countries were in a yellow book called the Mini Ucker. It was a physical book, and you wrote a letter to all of the people to say, I would like to do a PhD in your lab. I briefly toyed with a project that aimed to genetically engineer the smell of onions out of onions. And I also got offered a really prestigious, all-expenses-paid 1+3 position somewhere, but I failed to open the letter in time. So I have no idea where that was or what that would have ended up on. But so I ended up at Imperial, as Wendy said, in 1999 and haven't really left. So as a quick bit of audience participation, hands up who here is at Imperial. Okay, hands down if you've been here for less than 5 years. Less than 10 years? 15? 20? 25. So it's me, Nick, Ruth, and Shriley and Graham. So good. So proves the rule one thing. No one ever leaves Imperial. So if you're just starting your PhD, get used to it. You'll be standing here in 20 or 30 years' time. I studied a PhD in biochemistry, exploring the transgenic tobacco and vaccines with Gordon Dugan and Peter Nixon. Here we are looking a tiny bit older than we did at the time. It wasn't all smooth sailing. My 8 weeks of previous lab experience had not really touched the sides, including that most basic of skills of how to pipette. So I had to relearn everything from scratch. Peter's not here, so I can say I came in one day and he'd written across 2 pages of my lab book, "You are a shambles." Nothing else. To be fair, he was right. I was trying to purify this thing called Rubisco, which is the most abundant protein in the world, and I just ended up with a mess and no protein. So fair enough. Don't do protein biology. It led to a few heated exchanges about my competence or lack of it, and he was basically right. 6 months into my project, nothing was really working, which ended up with me moving to New Jersey to the Waxman Institute at Rutgers University. I was hosted for 6 months by the Cantor family, friends I'd made during my last trip at Project Use. And just to acknowledge the kindness of them in doing this, Philip, the father, pictured in the middle here, has recently passed away, and he was a wonderful man, and he gave so much time and space to me. I was working in the lab of Professor Paul Malaga, who was at the forefront of plant gene engineering. It was a very different setup to the UK. A lot more time was expected in the lab, but I did have one outlet, which was rugby. And this was probably the pinnacle of my sporting achievement. I represented Rutgers University, student body 50,000 in their first team for rugby. There's a bit of a caveat. Of those 50,000 students, 14 others played rugby. So my chances to get onto the team were somewhat elevated. And as you can see in the photo, our sports drink of choice was not exactly performance enhancing. On my return from the USA, I established my most important collaboration. I met my future wife, current wife, Charlie Weller. Future then, current now. That went well. I cannot stress how inspirational, influential, and wonderful Charlie is, and the enormous debt I owe to her. There is one other love in my life, the army. For 15 years, I was a reservist, juggling military training with science. I think it's helped my science enormously. It provided resilience, a bit of context where things weren't working in the lab, and an extremely colourful range of swear words. I like to think it helped my leadership style, though not everything translates. There was a time that I encouraged Katja, who's here in the audience, to JFDI. Look it up. There's a 30-second delay, but even though I don't think they want to bleep that out. And then she didn't speak to me for a week about how to sort that one out. So maybe don't use that one. My army career did eventually end, and it was a real wrench to me, leaving that aspect of my life. However, in more recent years, I have resurrected the old flame, speaking at World War II history festivals and marching with other veterans of unpopular wars. And it has led to extraordinary things happening. I certainly wouldn't have predicted 5 years ago that the pub landlord would be sending me research papers about dentists at the Battle of Arnhem. So what did I actually do in my PhD? And this is where I'm going to borrow from the Desert Island Discs format, which, if you don't know, is where a celebrity, me, I'll pause for you to absorb that, gets to choose 8 records to take with them if they were stranded on Desert Island, and a book and a luxury. But rather than records, and those with sharp ears who turned up early will have noticed my record choices, I've chosen the 8 papers I would take with me, and starting with this one, Expression of Tetanus Toxin Fragment C in Tobacco Chloroplast. And if you want to read along at home, you can scan the QR code. What we set out to do was make a transgenic plant that can produce a vaccine antigen. The idea was that by growing it in a plant, you can reduce production costs and potentially improve access in low and middle income countries. We used tobacco plants because they're relatively easy to regrow from a cutting. And you know what? It really worked. If you look on the big picture on the right-hand side, you can see the kind of yellowy leaves in the top left-hand plant. And that's because the plant is making so much of the vaccine antigen. It's basically killing the plant. So it was really successful. And we were the first ones ever to show that material grown in the plant could be used to immunize mice and then protect them against a lethal infection. I moved on, but the field continued to grow. 25 years later, a phase 3 clinical trial, so these are the big ones that go into humans, demonstrated that plant-derived influenza vaccine was as effective as that from a more conventional source. So from small seeds grow mighty oaks. Having solved the problem of vaccine manufacture for future generations, I was left with a bit of a quandary. What the heck was I going to do with my life? I proceeded to apply for a wide range of jobs to which I was completely unsuited, failing both the Foreign Office and Civil Service IQ tests. I was beginning to panic somewhat. And it was at this point that Tracy Hussell, who helped me learn immunology as part of my PhD, introduced me to Peter Openshaw. Sadly, Peter can't be with us tonight as he is sailing, but hopefully there's enough signal on board for him to be listening online. Tracy must have been quite convincing, as I really knew no immunology when I started my postdoc. Plus ça change. And so having done an apprenticeship in science during my PhD, I plunged in as a full-time scientist. So let's all now do an experiment together to start with. It relates to the research I do. I want you all to take a deep breath in. You just breathed in the following. About half a litre of air. You can breathe out, John. You do this about 20 times a minute. If you condense all of your breath over a minute, you can recover about 100 microliters of fluid, or about 1/100th of an ice cube. And then if you grow what you recover from that fluid, you get between 100 and 10,000 bacteria back. So every time we breathe in between 10 and 1,000 bacteria, and a surprising amount of skin. And the skin you breathe in mostly comes from people's feet. That's important. Not my research, but I think fairly important to know. And the question is, why aren't we sick all of the time? And the answer is immunology. The simple answer is the immune system. But there's no simple way to explain the immune response. It's multi-layered, and it stops us getting sick from infections. I really like this analogy that came out during COVID, which is like the Swiss cheese analogy. If you line up enough slices of wholly cheese, eventually there won't be any holes and things will be protected. There are many different slices, and vaccines are a really important way of adding extra slices in the way. One of the things I'm not going to be able to talk about tonight is the type 1 interferon response, but Dave Buss, who did a PhD in my lab, was exploring aspects of this in response to certain viruses. And one of the pathogens you potentially just inhaled is RSV. RSV is the virus that, since working in Peter's lab, I've been the whole focus of my lab's been working on. So it's called respiratory syncytial virus. It infects babies, and it affects everyone. It makes babies and the very old sick. If you go to the pediatric intensive care units in a hospital during the winter months, most of the babies in there will have an RSV infection. And it's very small. What I'm showing here is the genome. It's quite a simple structure, and this is the thing that we've always been working on. I have dallied a bit in bacteria, so Simran, who's in the audience here, explored Pseudomonas, and Sophie was working on Acinetobacter baumannii. I pretend that they were equally as interesting, but it's really all about RSV. I can say that now, Sophie's left. Which brings us on to my second paper. This was my first author paper from my postdoc. For those of you not versed in the weird system that is academia, papers normally have several authors, which can stretch into the hundreds. One physics paper has 5,150 authors. 24 of the first 33 pages are author names. The trick in biology is either to be at the front of the list, called first author, or, guess what, at the end of the list, last author. First tends to mean you did most of the lab work. Last means you did most of the thinking. One's perspective changes. So when you're the first author, you assume you did all the work. When you're last author, you know you did all the work. As someone who's now more often last than first, I can confirm my confirmation bias that I'm actually right about this. In this paper, we looked at the after effects of RSV infection. The idea of post-viral sequelae have gathered a bit of press coverage in the recent years. And it appears that most respiratory viruses, if severe enough, can cause long-lasting changes to the immune response. And so long before long COVID came long RSV. Infants infected with RSV are far more likely to get wheeze and asthma later in life. So this big panel above me here, you can see these are children who have bronchiolitis, and you can see out to 7 years, they still have a wheezy phenotype after they've had the RSV infection. Children who didn't have the bronchiolitis as kids don't have that. So it's causing some lifelong changes in the lungs. And building on observations from Fiona Cully when she was working with Peter, what we observed was that if baby mice were infected with RSV, they were not protected against reinfection. So you'd expect that an exposure to a virus once would protect you against future infections. And actually, in this age group, or in this animal model, the pre-exposure to the virus actually made them sicker when they saw the virus the second time. And what we observed was that this was associated with a particular type of immune cell called the CD8 T cell, which I'll talk a bit more later. And if we took this away, the mice no longer get sick. So basically the immune response had trained them to get sicker rather than less sick. I then had a brief interlude when I moved to South London. I can only put this down to sleep deprivation, deprived madness caused by having 2 children in 3 years. It was also where I first started working with Robin Shattuck, and have been working with him for 17 years. It's thanks to Robin's guidance and mentorship that I'm here talking to you today. A quarter of my academic output has been in collaboration with him, and amazingly, we still share labs and even speak sometimes. It's been great, and I'm completely grateful to you for appointing me and supporting me through my career. The first 3 years at George's were basically just me in the lab, but it led to my last author paper, which followed up the theme from above, but hasn't quite made it to my desert island list cutoff. And then in 2011, Robin was headhunted by Imperial, and as part of a buy one, get one free deal, offered me a job with him here. In a sliding doors moment, I moved back to Imperial and haven't looked back. It was where I appointed my first lab group, split over twin themes of RSV, immunology, and vaccines, that I've kind of worked on ever since. It was very hard to choose a paper from this period. It was really exciting setting up my group and kind of starting on the work. And in the end, I've chosen this one. In part because it continues the story, and because it has a useful lesson about guiding research. As you can remember, what I was just saying was that sometimes the lung immune system gets trained in the wrong way. So the CD8 cells, these are the T cells, these are part of your immune system that remembers and sees things, can make disease more serious. But other times, most of the time, the CD8 cells are protecting you against infection. And there's a particular type of T cell called the CD8 or resident memory T cell. And these are ones that live in your lungs. And they basically, once they've seen a virus, they're already in your lungs and they can fight it straight away. And so we, or specifically Katya, was able to show that the CD8 cells from the lung can protect infection with RSV. The important lesson in mentoring came from the experiment with which she demonstrated this. She had what I thought was a crazy idea of washing the lungs of one animal out and transferring the whole lot into another animal. I told her bluntly it would never work, but being somewhat stubborn, she went ahead and happily proved me wrong. So the lesson is, don't always listen to your boss. Sometimes it's just worth experimenting. The years at George's and early years back at Mary's were characterized by one other thing, the arrival of our children, Jamie and Sophie. I've lost my thread. These wonderful agents of chaos have enriched our lives in so many ways, and of all the things I've done in my life, being part of theirs is the one that brings me the most joy. Which is quite enough emotion. Back to the science. And I've slightly cheated here, linking up 3 papers, but the one I'm going to talk about is one by Helen with some input from Z. It's not actually Helen's poo. Just so her mum's sitting next to us, not too worried. As we've seen so far, CD8 cells, so what I've been saying is that CD8 cells can contribute to disease after respiratory infection. And one of the questions we've always wondered is, and the way we measure that, is that mice lose weight when they're infected. So what this graph is showing is that they initially, if the uninfected mice don't lose any weight, but if you give them RSV, they lose some weight. If you take those T cells away, they stop losing weight. And what Helen asked was why? And the really simple answer is they stop eating. So and you'll notice from having a viral infection yourselves, it suppresses appetite. And we actually were trying to pitch RSV as like a miracle diet cure, but Ozempic beat us to it. But having seen this, Helen then made a follow-up observation, is that the gut microbiome, so the bacteria that live in your body, also change when the viral infection, because people stop eating, the bacteria that the people carry around with them also changes. So there are massive shifts in this gut microbiome that's driven by the thing. And then there was a follow-up study, Z, who just left us for La Jolla in the US, with good luck with that timing, found a signaling cascade. This links a cytokine released from the lungs that enters the brain, leading to hormones relating to satiation being released, reducing appetite. So there's a whole circuit of basically lung to brain to appetite to back again. And why this has evolved, we don't know. And trying to kind of close out this question is the next step in this area. But it's really fascinating. And that was a collaboration with Miriam. So thanks for all the help on that. One aspect of my work I've yet to acknowledge is the brilliant collaboration with all the companies that over the last 15 years. These have always been interesting, and experienced science closer to the kind of coalface of products adds a different dimension to the work. I'm going to single out a single partnership, and that's with Touchlight Genetics, with who I've seen grow from 5 people in a shed in Leatherhead to an enormous world-first DNA manufacturing complex in an old sewage works in Hampton. Being part of their journey has been a joy, and it certainly helps that they've hosted some absolutely fabulous parties. Speaking of industrial collaborations leads us on to Desert Island Paper number 5. In 2014, I was approached by Steve Rees from a relatively unknown German company. The company was called BioNTech, and at the time, they had 4 people in their infectious diseases arm: Steve, Annette, Steffi with an F, and Steffi with a PH. We set a collaboration funded up by the EU to investigate their platform, self-amplifying RNA, which we're looking at. And what you can see on this one is that this is measuring, the HAA titer is a way of measuring antibodies. And what you can see is that the right-hand graph, all the numbers are 100 times lower than the left-hand graph. So this vaccine, you could put in 100 times less and you get protection. And actually, in work that David Sterling did as part of his PhD, we brought this down to 10 nanograms, which is almost homeopathic levels of RNA. But what became of BioNTech? On the back of this, and admittedly a lot of other data, they started a partnership with Pfizer, made an RNA vaccine for COVID, and from there, history was made. So one of the take-homes from tonight is big outcomes can start as conversation in fish and chip shops. Figure 6 draws on a similar theme to paper 5. Figure 6, paper 6, RNA vaccines, that comes with an even better backstory. As part of our collaboration with BioNTech, we started some pilot studies with Kati Kariko. And Kati is very much a poster child for the ups and downs of science. She fled communist Hungary as a girl with $1,000 hidden in a teddy bear. And when we first met, she came and gave a talk with just 30 people at Imperial. When I tried to speak to her last, she was off to meet the King of Belgium. The idea that she worked with, Drew Weissman, was that the immune system has different ways of recognizing RNA. And what this figure is just showing, there's different types of RNA. And if you change the RNA, you change the way the immune system sees it. And we worked with her, and we saw that if you put in the modified RNA into our system, you get a different gene profile, and then you get better immune responses. But back to the backstory, we had that first bit of data during a collaboration that ended in 2019. And come COVID, my lab had kind of shut down, and I wasn't sure what to do. And I was just going through uncompleted projects. I remembered this, and with mRNA being the hot topic of the day, I spoke to Katy, and we finished the rest of the project. It led to two great things. First, my first journal cover image, and secondly, a Nobel Prize, well, sort of. Not mine, I should stress. The paper came out on a Monday. The Nobel Prize was announced on the Tuesday. So until she'd published with me, Katy was not a Nobel Prize winner. And it's worth noting that causation and correlation do not always equal. So that's the briefest of run-throughs of some of the science I've done on the way to Professor. So what have I learned? T cells have a contextual role. Sometimes they protect, sometimes they enhance disease. The immune system is quite complicated, and I think this is the most important thing. If you think you understand it, you don't. And I think that's really important about biology in general. But if people tell you it's simple, it isn't. And the people who are like, oh, AI can fix everything, are wrong. So there is a lot more to be done. Luckily, I have 20 more years to work some more of it out. But crucially, vaccines work, and we can now make newer, better ones faster than ever before. So you may be thinking you're getting close to the end, but don't worry, there's two more parts. In the winter of 2019, we started to hear about the outbreak of a respiratory virus. And at this point, I'd worked for 15 years on respiratory infections and vaccines. Now, if ever, was the time to make my mark, and I did. I built an enormous, and some might say record-breaking tower of Lego with my daughter. I went for runs with my son, who's now faster than me, and I baked some terrible sourdough. But importantly, I got to pause, and I got to rediscover writing. My first writing output from the pandemic were the COVID diaries. This was a collaborative effort between myself and Jack Leeming, who's the careers editor at Nature. It was tremendous fun. Every week I'd be thinking up one idea, writing the next, editing the third. We covered such vital topics as houseplants, homeschooling, wearing slippers. Even better, I got paid and used the money to buy vital things like a sausage maker and a toilet roll subscription. But best of all, it all went onto the big science indexing site called PubMed. So at one point, I had 10 Nature papers in a row. And I reckon my 26 articles in that esteemed journal puts me near the top of the Imperial academics, by number, if not necessarily impact. My dalliance with this weekly column led me into thinking about more writing, and it really lit the thing. So I have always enjoyed writing, and I wrote for my college newspaper as an undergrad. I've actually written for Felix, the Imperial magazine. About 10 years ago, I set up an academic blog, which no one read, but it led me to writing for Times Higher Education and then for Nature. But importantly, this then all led to my two books, which you'll be impressed that I've not mentioned till now. If you've not heard, they are available in the foyer after this. And none of this writing would be possible with all these fabulous people. On the top row, there are people who kind of helped me with the magazines and the writing. So Nick, Alan, Julie, Jack and Paul, who all put me on the path. And then into the world of books, we've got Dan Davis, who's here, whose advice still sticks in my head. He said, write whatever you want, just don't make it rubbish. But it was a shorter word that rhymes with hit. Caroline, who's our agent, Sam, who's in the audience, who's the editor here, and then Ben, who helped with some of the narrative on the new book. Which brings us to my last Desert Island paper. And that's the one that's just around the corner, the one I've not yet published, the shining light of excitement. As I wrote this, I was writing up some of the papers, and it's a process of like akin to sculpting. You often have this pile of data, and then suddenly something clicks into sight, and it all makes sense. Hattie, who's in the front row, was doing a paper about a way of modulating delivering RNA, and nothing had made sense, and then suddenly she developed some RNA data, and it was like, oh, that's what it is. And the story all suddenly dropped out. And it's so exciting. I love pulling the stories together when it all works. And so for those of you au fait with the Desert Island discs, as well as the records, in my case papers, you get to choose a book. This is an almost impossible decision. And I'd obviously encourage anyone else in the same situation to take one of mine, conveniently available in the lobby. But I don't need to hear my own voice. So my choice would be A Day in the Life of Ivan Denisovich by Alexander Solzhenitsyn, which is all about finding joy in small things in difficult circumstances. But you also get a luxury. So rather than the luxury, I'm going to take a liberty and share some lessons learned with you. And for me, the main lesson of a career in science has been the how matters as much as the what, and particularly the people matters. And I think my science legacy, I hope, is as much the people who I've trained and worked with as the kind of science that comes out. And conveniently, I can group them into three groups. Category 1, if you make me touch a pipette again, I'll scream. There's quite a few in that group, I'll admit, so maybe I didn't get it quite right there. There is more in category 2 of, in spite of everything, I think I'll stay in science. There is then one final tricky category, which is, I'll continue to do science, but only if I can work for your wife. I'm not sure how to interpret that. The two of them are here, so maybe it's not all bad. Beyond the legacy of people, here are my 6 other tips for a happy and prosperous career in academia or in science. Conveniently, they all begin with the letter C. I tried to choose the letter J, but even ChatGPT couldn't come up with 6 synonyms for these ideas. It did suggest jazzy and jocular, which are pretty terrible words. So my career tips are be curious, creative, communicative, collaborative, comedic, and collegiate. Even that was a bit of a struggle. So first one, be curious. Stay in something long enough, and eventually it becomes relevant again. I'll just give you an example of RSV. So I've been working on this RSV virus for a long time, and there'd never been a vaccine until 5 years ago, and suddenly this vaccine came out, and I actually thought, oh no, that means it's the end of the story, and it's the beginning of the story. We're looking at how the vaccine interacts with the protein, how the antibodies might be changing. Valeria, who's here in the audience, and Barbara and Paul, we're all looking at the different structures and how things change, and it's so exciting. It's a really exciting project. I don't understand how Barbara and Valeria do it, but they can work out how the two proteins talk to each other at every individual contact point and then mutate them and change them in silico, and it's brilliant, and it's so exciting. So just being curious about things and staying interested in ideas has been really important. I've really enjoyed that space. Be creative. I can speak for an hour about this, but science is a creative subject. If you're at the point where you're deciding where you're going to go with your career next, and you're thinking about, oh, but I'm creative, I can't do science, or oh, I'm only doing science because I like doing sums. Science is creative, and I think sometimes we split people artificially at 16 into these different disciplines, but actually a lot of the time I spend now is thinking about things and how to make things work. And if you want more on this, my friend Richard, who's a cartoonist, who drew this brilliant my favourite cartoon about mice, he sells greeting cards, but we wrote this whole article about our creative processes together, and actually we share loads of overlaps about the way things work. So it's been a really interesting, and I really enjoy that aspect of science. Now, this may not be the most standard advice you hear, but if there are 2 options and one of them's funnier, that's the one to take. There's a danger of people taking themselves far too seriously in science, and there are many opportunities for humour. Some simple things are saying toot really loudly down a cardboard tube into people's ears in the lab. Tom might remember that. Others are slightly more subtle, like getting Schrodinger's cat's bottom into nature. And others have a longer payoff, like changing your friend's name Steve to Steve-O in an academic paper. The best thing was, I'd forgotten I'd done it, and the first thing I got was a text from Steve saying, "You swine." He used stronger language because he's from Essex. Academic life is more than just doing the research that we want to do. There are lots of other things that need to happen for the whole thing to keep turning. We all have to take on our share. I think Bill and Ted put it best: be excellent to one another. And you've got 2 more to go. I'm pretty much on time. Science shouldn't exist in a vacuum. I think, Dr. Strangelove saying the whole point of doing science is lost if you keep it a secret. If you work on something that's brilliant, but you never tell anyone, there's no point in doing the science in the first place. And I really enjoyed the different ways of communicating my science. I enjoyed talking in this environment. I enjoyed writing papers. And I think that's something to build on as a skill. And finally, I've made many, many wonderful friends in science, many of whom are here tonight, some sadly who couldn't make it. Alice and Tom and Ian and Rob and Sean and Cecilia and Steve. They're people to drink beer with at conferences, shoulders to cry on, people to laugh with, and particularly a sympathetic ear to slag off other academics too. And so I'm going to steal... Oh, no, hang on, I need my phone. Can I have my phone back? My dashing assistant. My future wife, no less. So, to steal the punchline from my own book, If you take one thing away from this talk, it's this. Don't use that to make the slides work. Do that. Do more of what you like with people that you love, and you'll enjoy it. So I'm going to take a selfie of us all. I'm going to take it off the wall of everyone having great fun here. Thank you.
Well, thanks, John. That was wonderful, and I'm very glad you gave us permission to laugh, because otherwise we would have all been suffocating trying to stifle it in. It was a smile from start to finish, and a wonderful journey to go on with you from your career to all the things you love doing. I do wish that we could have brought your cardboard cutouts inside so we could have played with them a bit, but I'm sure they're available after the event, like your book. I'm open for questions. Paul?
It's not a science one. What's more relaxing for you? Presenting your science to an academic audience or talking about your book in a book festival? And why?
The science, actually. I think I've certainly found with the talking, the public talk takes me a couple of hours to build myself up to it and then an hour to come back down from it, whereas the science talks I don't have to pace up and down as much. So I think probably the science, probably just more practice and doing it, and you're doing the same talk maybe a bit more times.
So more books then? And then
more books, and then, yeah, exactly. That's the hope.
Shirani?
Hi, John. During COVID, a lot of us enjoyed reading your tweets, as they then were, I believe, particularly your attempts to make bread and so on, and your retort that you couldn't do PCR in your kitchen, so why on earth should you be bothering, reviewer one, etc. Obviously, a lot of us kind of engaged in social media to an extent, lesser or more. Do you think it's kind of gone wrong? And what's your advice to the rest of us about how to use it now in the era of kind of X?
Yeah, I've stepped back from the science side of it. I still use it on the military history stuff. I think it's really enjoyable. There's an active community of nerds who share stuff, and people I've met online and then in person and then online again. That bit I enjoyed. The dipping a toe into the fire pit, I can't face it. The people on there are so angry about everything. They're wrong about most things. It just feels, it feels too hard. So I think as long as I think the better thing is to provide information in different ways where people can access it. I know OVG tests it. They have a brilliant website where it's really clear about how vaccines work. And I think probably in a less confrontational way, it's better ways to do it. And I don't have the energy or strength to deal with that kind of level of stuff.
One at the back and then one at the front.
You can see, I'll come. I've got to get my...
Follow the hand sign.
Hello, Nigel Reeves, uncle by marriage. As someone that didn't finish their PhD, and you freely admitted that you had a difficult year, the first year, was it just the fact that it started to work and you got those results that made you sort of hang in there and continue and complete your PhD?
Yeah, I think so. I'd gone in with a completely naive opinion of what a PhD was going to be. I thought, I'll just be another undergrad for 3 more years. Week 2, when I dialed in saying, I'm a bit hungover, I can't be bothered, I again got a slightly large earful from the supervisor. So changing my perspective to understanding it was a job and that you have to work quite hard for it. No hint here. That helped, but I think if it carried on not working, I would have really struggled with that. So I think that the tipping point was things beginning to work again, and the opportunity to go to America was extraordinary, and it really did help.
I've got a question at the front. Don't know who.
Cousin by marriage. Or just by cousin.
Thanks, John. Great talk. As someone that works in the lab and has obviously never put a foot wrong, I was wondering, what is the first thing that comes to mind of the biggest thing that you've messed up in the lab in your career?
Just need to look. It wasn't here. It's the first thing. Sharon's not here, so that's good too. There are quite bendy pipette tips. There's a certain type of bend, the long ones, when you're loading a gel. And I flicked one, and it went and vaporized across the whole room of whatever nasty thing it was. And it took me an hour just to scrub it. I locked the door, scrubbed it, put it in some bin bags, and never told anyone. But it wasn't here.
That's a relief.
But it was hot in the mouth. It was, oh, God, what have we done?
Any online that I need to be aware of? Can I, oh, there's one here.
I just want to take a little word about the pickled onion gene, which I began to study aged 19 when I was ill in Cambridge and got better by wandering around a supermarket and found the only food which made me better was pickled onions. You say things come back again. Do you think at 78 I should start again researching this more seriously and go for a grant?
In the vein of more of a comment than a question, yes.
Excellent. A question at the back?
Yeah, hi, I'm Taran. Thanks so much for the lecture. Just looking back at your younger self, what was your younger self? What was one thing that he was most correct about and one thing that he was most wrong about being a scientist?
Most correct. You have to be very good looking. No, I didn't understand science as a thing until I started doing my PhD, really. I think I enjoyed reading about it. I enjoyed the ideas, the stuff you're being taught, the process, but I had no clue what it meant, how difficult it is, how the thing I really enjoy about being a lab head is that it's no longer jeopardy all on me. I've now got 5 people whose days can all go terribly, but at any one point, one of them, stuff will be working. And you take yourself away from that every day of being a PhD student. You're like, oh God, nothing's worked again. And so I hadn't appreciated any of those aspects of it. So I didn't have a clue what it was. I had no idea what I wanted to be till I was 30. I'm really happy with where I've landed, but I don't think young me had any clue. It's a bit of an admission. I'm sure I was right about some stuff.
Thank you. Hello. Yes. I have just currently taking your online course on Coursera about vaccine, and I also have watched a YouTube video about the lecture of the Royal Institute. I wonder, how do you balance your time between writing books and the teaching?
Yeah. So with a lot of sympathy from my wife, the Coursera course was organised by Sophie sitting behind you, so it was an official piece of Imperial teaching, so that counted as my contribution to the Faculty of Medicine, Deborah. The books I write at weekends. In COVID it was easier, I've been told, because there was nothing else to do. The second one maybe got in the way of me doing any form of parenting, which I'm slightly guilty about. So it's a juggle, it's a juggling act, and finding space. We're very privileged as academics to shape our own structure of our weeks and what we work on and what we choose to work on. And as long as we're teaching and supporting and helping everyone, there is space to do your own things as well. So it's a very privileged job.
Miriam.
John, I've had the real pleasure of collaborating with you, and you've really highlighted the importance of collaboration and teamwork, and what I really admire is the meticulous when you're doing experiments, covering everything. And I think you've also highlighted how you've really made some discoveries that then have really launched the next step of things that we know about. So where are you going to go next with your science? What sort of egg would you like to crack next?
The one that's the most interesting. I think what's really nice about preclinical early stage stuff is you have no idea where it's going to end up. So I couldn't have picked where the RNA was going. I couldn't have picked where the vaccine, any of it. I don't know where it's going to end up. So I think where we are fortunate in this country is that MRC welcome fund us to do this blue science basic stuff that then builds into who knows where next. I said to Dad 25 years ago, the thing that I hope for most is that you might change half a page in a textbook. And maybe it's different to that now. It's more about, have you put one building block into the tower that then changes everything? So the thing I'm really enjoying at the moment is this RSV, the antibody stuff, because it's so interesting, so fascinating. But I'm really lucky to have the co-work with Robin on the RNA vaccine. So we've got these two strands, which I find fascinating. I had a dream about RSV the other day, which is maybe oversharing. But yeah, so those are the things I do. So it's always been things I enjoy. And it's about tinkering and taking things apart and trying to understand, peeling back the onion skin to find the next layer, and it's more complicated.
I think we should maybe end there with the onion analogies. They seem very appropriate for this evening. And I'm going to hand over to Robin to wind us up.
So good evening, everybody. I'm going to start by saying, because I'll forget otherwise, once we finish, there's a drinks reception afterwards, and you're all really welcome to join. And in fact, I think we'll be disappointed if you don't join us to celebrate informally afterwards. It's a real privilege to be able to stand here today and offer a few words of thanks after such a thoughtful and entertaining inaugural lecture. I've had the honour of working with John for 17 years, and I've learned lots of things tonight. One thing I learned is if I had known what he said tonight, I would really not have wanted him as a PhD student. But it's been a genuine pleasure to witness his journey from his first academic appointment all the way through to becoming a professor in his own right, a very deserved promotion. You will have heard from the talk John's immense passion and enthusiasm for vaccines, for immunology, for RSV. I don't know how many times I've heard him talk about RSV to me, RSV, but it's really paid out. And that's really a part of who he is, but you haven't heard all aspects of what he does. And really, he has a remarkable talent for communicating science, particularly in the written word, popular science. And I think one of the things he's shown that he's really passionate about is not just doing research, but actually telling people about research and having that connection, that ability to communicate with the wider public is becoming increasingly important as we move into a generation that's becoming more and more skeptical about what science does and about what real truth is about. And I think John is really accomplished in doing that. I've no doubt that we'll look forward to more books, more blogs, more tweets for many years to come. And I look forward to seeing what will come out in the future. But John also inspires a great deal of warmth, respect, and appreciation from everybody who works with him, particularly his students, past and present, who are here today, many of them. And I think that's testament to his skill as a teacher and as a communicator. And all of that, despite the fact of his really bad taste in jokes. Within the college, John has been an advocate for equality, diversity, and inclusion, a champion for mental health, and a strong supporter of innovative teaching, things that many of you may not know unless you're working closely with him. And those are also really important parts of his career development. I'm surprised that he takes such a genuine interest in all staff, in all research, and is a great encourager of everybody at every level. And that's a testament to his generosity. What's particularly inspiring, and I think is really nice to note, is that he's forged his own career path, very much a very balanced career path. Many academics are very biased towards one aspect, teaching or research or communication or doing college responsibilities. And John managed to blend all those different aspects together. And I think that's a testament to him building a career that's very much his own. And I hope many of you, particularly those who are starting out in science, serves as a reminder that a career path really is a unique journey, and that there's a value in embracing absolutely what's right for you and not necessarily what you perceive as the only path to success. And success comes in many different guises. It isn't just about academic progression, which John has done, but it's also all those other aspects that make you a rounded scientist or whatever discipline you're in. I think John was also very good in kind of illustrating the ups and downs of science. And an awful lot of science is actually about the downs, and we're rewarded by those occasional successes. And John shows an immense ability to bounce back. Usually it's about 24 hours when something goes wrong, and then he's coming back with enthusiasm. And that resilience is really important. Now, some of those things are innate, some of those things are learnt, but I think that John would want me to say that much of that is strengthened by the support, particularly of family, friends, scientific colleagues. And so I would want to thank all of you for supporting John through his career, whether it's for a long time, whether it's his family from his birth, whether it's colleagues collaborating over many years, or whether it's just turning up here today to celebrate his inauguration. So I would like to thank all of you. And finally, I would like to ask you to join me in thanking John for such an engaging and inspiring talk. Book purchases, of course, are optional. But applause is not. So I'd like you all to join in applauding John for his wonderful inaugural lecture.